Good morning, everyone, investigators, researchers, or faculty students. Today we're going to present a seminar about the facilities or core facilities of the new RCMI program that was renewed recently. Actually, it was renewed in September 20, a day we never forget. It wasn't that bad after all. So that means that this program was renewed for the next 5 years, and instead of each individual core facility, all core facilities are united into one research infrastructure core, but with the same directors of each individual core. And so we, in addition to counting with the RCMI program, we also have support from other grants like INBRE program, the Cancer Center, and the PRC for some of the resources. So starting, we're gonna present the presenters, starting with me and the director of the Proteomic Center, followed by Dr. Carmen Cadilla, the director of the Genomic Translational Research Unit, Natalia, the director of the Metabolomic Core from INBRE program, and Adelfa Serrano, the director of Tropical and Emerging Infectious Disease Core, Mark Miller, the director of the Neurogenetics and Bioimaging Core, and Abiel, the director of the INBRE Core. Bioinformatics is one of the component of the core, and he's the director. The outline of the presentation follows here, starting with speaking about what components comprise the RCMI Research Infrastructure Core that we're going to abbreviate as RIC, including proteomic, metabolomic. Metabolomic is in addition, is from INBRE, but it's also, we're going to talk today, genomic, emerging infectious disease, molecular neurogenetics, and informatics. We're going to speak about the aims of the new grant, the mode of operation, and then emphasize the acknowledgement that we have to provide, which is now a U54 grant. And every time we do a presentation or a publication, we have to remember to include this little number. And everything is going to be available in the website that is going to be also, it's being revised as we speak, updated. So this summarizes the different cores, like for example, in the medical science campus, we have the genomic service, the tropical emergent disease, infectious disease, the integrated informatics service. In the Institute of Neurobiology, we have the Neurogenetic and Bioimaging Services. In the Puerto Rico Comprehensive Cancer Center, we have the Proteomic Services and the Genomic Services that also collaborate into a group called Promix within the Cancer Center. So everything now is like integrated. And the aims of the new research infrastructure core facilities are to combine bioinformatics, health informatics, operational informatics resources into one integrated informatics service, to deliver high-end service to support genomics and proteomic research, to provide state-of-the-art neurogenetic and bioimaging services, and to provide access to key resources for tropical and emerging infectious diseases research. The method of operation, every time we use these facilities, we have to consult with the director for experimental design before we bring them a sample. We want to make sure that the samples are appropriately collected, participate in helping you guys in the sample preparation, the quality assessment, and in the design of the experiments. We're going to contribute to data analysis, validation, and even to the presentation and publication as needed. The initial consultation visit would include discuss of the hypothesis, the research questions that we have, the objective of the researcher, explore the options that the person wants to have in omics services, and look at the literature review to make sure that the questions are not something that is available already in the literature. We want to help you guys to publish, to get new things, to innovate. So it's important to look at the literature, discuss collaboration fees, and also sign an agreement to make sure that all the parts are in agreement in terms of the participation, and also even to the level of the publication. Discuss user guideline and follow or file a registration form. include assurances, so make sure that if you're dealing with human subject, the IRB, if you're doing with chemicals, with the biosafety, etc. And have a timetable. For example, when do we expect your samples to be brought to the core, so we can calendarize or set your experiments ready when you are ready. So we're starting with the Translational Proteomics Center, which is the one that I direct. We have the staff in the new grant is covered by Carla Salgado. Carla Salgado is a chemist with 10 years in mass spectrometer. She used to work with forensic science, and she's been with us 3 years. And she actually is the main operator of the mass spectrometer. Both mass spectrometer, we have two now, one for protein identification and one for protein quantitation. And we also have Yadira Cantres. She's a biologist, but she has 3 years with us in the core. They're both sitting here. Can you stand up? And she's the research technician. She also is in charge of all the instrumentation in the core, and she also helped in sample preparation for proteomics, and work together with Carla. And in addition, we also collaborate with Horacio Serrano, who has preparation in quantitative mass spectrometry, and he worked with Eric Miranda, a research technician that he's also a statistician. He has a master's degree from public health, and both are help from internal medicine. So internal medicine has provided help to work together as a team in proteomics. And we also contribute to the Promix in the Cancer Center. So the goals of the Translational Proteomics Centers are to provide an integrated platform for biomarker discovery and protein ID and quantitation to interface between basic and clinical research, conducted in Puerto Rico for the diagnosis and therapeutic monitoring of human diseases. We also want to integrate with the other cores, the genomic, bioinformatics, metabolomics, bioimaging, and emergent infectious disease in the proteomic platform. So whatever we can do to work together, publish together, we're going to emphasize. We have meetings every month. We want to also help the postdocs that are working in this area to grow them into scientists of the future. We also want to provide service, because the emphasis of the RCMI now program is to provide service. So we provide service with new methodologies on quantitative proteomics, such as SILAC and TMT labeling. And we provide validation strategies, including imaging in collaboration with neurobiology, ELISAs in collaboration. We have available instrumentation, but also the emerging infectious disease facility, and kinetic assays and flow cytometry in collaboration with the Cancer Center. They have an instrument available. We can also help you with that. Provide workshop and training, but this is through the INBRE program, because this is an objective of the INBRE program, is to provide workshops for the INBRE investigator. But when we do that, we also open that to all the investigators that need this specific technique or for their experiments. And this is the organigram of the RCMI Translational Proteomics Center. As a director, I oversee the research associate, Carla Salgado, works with the mass spectrometer, and Yadira Cantre, that is also going to be supported, is temporarily being supported by INBRE, but it's going to be supported by the Cancer Center starting in January. So, and also we have collaborated on consultant. For example, we collaborate with Brian Balif recently. It's been about 2 years. He comes here in March to provide workshops. He has a grant that includes us to come over here or have students go over their facility in Vermont. It's an INBRE facility. We also collaborate with Paweł Siborowski in Nebraska. He's coming in at the end of January. So we have ongoing collaboration with them. And this is the scheme right now, the overall general workflow of the facility. We do now protein identification and also protein quantitation. For protein identification, we receive the samples. We could be, if it's clinical samples, then they have to be processed by the group with Horacio and Eric, that they do depletion of abundant protein from serum or plasma, but it also can come from tissue culture, cell culture, and it's important to do the extraction of the proteins by cleaning with acetone precipitation, do protein profiling by using also one-dimensional gel electrophoresis, and or if you're interested in protein-protein interaction, we can do immunoprecipitation to look at the protein that interact with your target protein, followed by the gel, and also then purify those bands by an digestion of the protein with trypsin. And it looks simple here, but it's a lot of steps before going to the mass spectrometer for protein identification with the LTQ. Then followed by analysis, and then we will give you a list of proteins with the statistics for what are the most abundant protein, more relevant proteins, so you can pursue validation of a protein by either ELISA or Western blots. In the future, we're going to introduce the SRM method for targeted quantitative proteomics also for validation. They're learning that also through the INBRE program that send them to do workshop in the United States. Protein quantitation, the same, we have to process the sample the same way, but also you can do, before protein quantitation, mass tagging, which is called TMT. And you can have about 10 different tags, so you can accommodate 10 samples into one sample before going to the mass spectrometer. So your charge will be for one sample of 10 samples. You know, all these prices are available in the website, after that you also have Bioinformatics. It's going to be collaboration with Bioinformatics group to do the analysis of all these proteins in terms of which ones are the more significant. And then you can also pursue validation by ELISA or Western. This is the list of services that I'm not going to go in detail, but they are available in the web, and also with the price for each one. And we have lowered all the prices so that they are something that everybody can afford, because I know we're going through tough times, and now that we can do it in-house, then we don't have to do shipping and all that and pay. Also, it's going to be much more available price. So in summary, starting with protein processing of the samples, protein fractionation, protein separation, protein-protein interaction studies, sample processing for mass spectrometry, protein identification. Quantitative proteomics could be iTrack or TMT labeling. We are promoting more TMT labeling than iTrack because it's most cost-effective and targeted to the current instrumentation. We also have available SciLab, which is amino acid in culture. So if you're going to culture cells, you can add already the amino acid that has different mass, and then you don't have to label post at the end of the experiment. It's already labeled. And analysis by the QX-active protein validation. We have available fluorometer, ELISA, Western blot instrumentation. This is a small flowchart of TMT label from a paper that was recently published, not by us, but by a group that studied cancer proteins. And they apply the TMT label and they validate using the SRM method. And it's a very interesting paper, include that references. We have recently worked a paper from our core, but it was in collaboration with people that already have the quantitative proteomic instrumentation. Now we want to do it in-house. These are the location of the facility in the cancer center. We also have this space available for workshop, but students can also work there if they need to work there together with the technician. We also have a chemical room, a cell culture room, and a cold room, and emergency outlet that helped us a lot during the hurricane, so we didn't lose anything, thank goodness. And the instrumentation, in addition to the basic instrumentation, we have an ultracentrifuge and weight balance, incubator. What else? This is for the gel dog, also for the Western blot, virus scan flash for the ELISA and fluorometry, and electrophoresis and Western blot equipment, and also for the protein identification, the LTQ, which now has also coupled thermo ECLC, which is this part was provided by INBRE. So the LTQ was provided by RCMI and the other parts. So now it's like updated instrument and it's working very well. And now this is the new instrument, the QX-active, which is we're already running samples for quantitative proteomics as we speak. This was provided by the INBRE program last year, and it arrived just before the hurricane, and it was installed after everything was normalized. In addition, we have analysis program. These are the program for the Western blot imager. You can do quantitation of the Western blot band using this program. Also for the VarioScan for the ELISA, this is integrated into the VarioScan Flash and the fluorometry. Also, these are for the proteomics LTQ, the Excalibur proteome discovery, and we also share with Bioinformatics the ingenuity pathway analysis that they're going to be running those for us. So the program is also being renewed for the next 2 years. And now we have, I introduce to you Dr. Natalia Chorna, who's the director of the Metabolomic Research Core, sponsored by the INBRE program and located here in the biochemistry department. Natalia.
Hello, my name is Dr. Natalia Chorna. I am the director of Puerto Rico INBRE Metabolomics Research Core, which is located at Biochemistry Department on the 6th floor, room A632. The long goal of Metabolomics Research Core is to increase the number of investigators which can be cross-trained in metabolomics by providing the access to modern state-of-art analytical tools and services. The aims of Metabolomics Research Core is to provide training, knowledge dissemination among researchers, undergraduate and graduate students in Puerto Rico, offer specialized supporting service for assay development and specific metabolites identification, and we support established a new collaboration that exploit metabolomics approach to move basic and clinical research toward translational goals. This picture shows the progress of Metabolomics Research Core network collaboration growth. Initially, the core was located at the, unfortunately, technical difficulty, was located in University of Puerto Rico, Rio Piedras campus. At that time, we were from 2011-2015. We established collaborations with 3 universities, 6 faculty. We trained 7 students, 1 technician. In 2015, the core was moved to University of Puerto Rico Medical Sciences campus, and we grow, and now we have collaboration with 10 universities, 1 in United States, University of Wisconsin Madison, 16 faculty. We trained 42 students, 4 postdocs, and 4 technicians. We have been productive during the time of establishment. We analyzed 1,500 samples, and each sample has from 30 to 60 metabolites. In total, we identify 60,000 metabolites. We provide 16 seminars and workshops. We supported 29 conference presentations, published 3 papers, submitted 2, and participated in submission of 3 grant proposals. The area of core current collaborations are neurobiology, immunology, bioengineering, genetics, bioinformatics, cancer, pediatric urology, and biotech. As an example, I can just give a few examples what we are actually doing, how we collaborating. In area of neurobiology, we are collaborating with Dr. Miranda from University of Puerto Rico Medical Science Campus. We identify biomarkers of severity of spinal cord injury and inflammation and chronic pain. Dr. Corina Bergen from University of Wisconsin Madison, we are identified the metabolic alterations that accompany increase connectivity within the brain of environmentally enriched rats, which they identify using MRI technology. In cancer area, we work with Dr. Godot Vitorina from Inter-American University, and we are using combined approach, metagenomics and metabolomics, to study microbiome dynamics in cervical cancer prevention. Another collaborator is Dr. Kucherevich from Universidad Central de Caribe. We actually completed this investigation. We identified that HIV envelope protein GP120 promotes glioma cells growth through glycolysis. This paper is currently submitted to Oncogenesis and we are waiting for revision. The other area is pediatric urology. We are working with Dr. Perez Brayford and Jorge Rivera, and we identify metabolic biomarkers of hypospedia severity. Also, the newest collaboration was established with Dr. Lopez Garriga from University of Puerto Rico Mayacoez campus. We are planning, using mass spectrometry, identify the heme products, different heme products. Also, we're working with genetics, in genetics area with Dr. Carmen Cadilla. That was the first our collaborative project. We are identifying lipid compositions in human embryo glands where transcription factor TWIS2 is manipulated. Also, we work in area of biotech with Dr. Baerga, and we are optimizing the conditions, trying to get the better production of polyunsaturated fatty acids in Escherichia coli. What is metabolomics? This is workflow, the first chart. We are using biological sample. It can be biofluids, cells, and tissues. The sample is undergoing extraction of metabolites. Currently, our facility based on gas chromatography mass spectrometry technique. So we can identify fatty acids, sterols, amino acids, carboxylic acids, and some others we are working on. Then we separate metabolites using these techniques. We collect data. And we perform bioinformatic analysis to identify biomarkers and affected metabolic pathways. You should realize the metabolomics can be used like retrospectively. When you identify affected metabolite, you can go backward and you can end it with a gene. So through ferments and activation and genes. So it's one of the powerful techniques currently. The technology has advantage over others. It's relatively cheap. It can measure up to 100 metabolites with no special separations before. It's mostly automated and requires some, of course, it does require some sample preparation and derivatization, but it's not difficult or expensive. And then it provides you instant identification of biomarkers and affected metabolic pathways using available resources, which I will tell you on the next slide. So this is the instrumentation of the core. We have GCMS and GCMSMS from the Shimadzu. Both instruments equipped with comprehensive databases from National Institute of Standards and Technology and Willy Registry. Together they have about 1 million mass spectra. So when we run our samples, we match the mass spectrum we obtain to database to identify with high probability metabolites which will be used further for biomarkers identification. When we collect our data, we are using the free resource Metabo. Sorry, I'm not familiar. Metabo Analyst. This is free-based tool developed in Canada. You can upload your data and perform any type of bioinformatic analysis, including principal component analysis, partially squared discriminant analysis, heat maps, and so many others. In addition, we are using Ingenuity pathway analysis to identify other components of the sample, the components of the affected pathways, and map it to known knowledge pathways like KEC pathway and others. So, thank you.
Bueno, ahora yo les voy a hablar de las facilidades que tenemos disponibles para... Oh, I have to do it in English. Sorry. So I'm going to be talking about the facilities that we have available for genomics research and genetics research also as well in our campus. Like Dr. Melendez and Jordan have mentioned, we are trying to share our resources and also provide a better picture of how to design experiments. So all of the courses have the same scheme, like Dr. Loida Melendez explained, which is if we advise you to come to us for the experimental design, and also in that regard, other people can help, for example, the Bioinformatics course or the Informatics course. But if you have a proper design, then you can get useful data that you can then publish. If you don't have an adequate sample size, then you probably won't be able to publish your data and have spent a lot of money to do something that might not be publishable. So in genomics, services, we started first as a molecular biology core, which then expanded to genomic services when I was introducing the equation. So we still retain and institutionalize the molecular biology core, which is still directed by Dr. Jose Rodriguez Medina, who started it all. And we have then maintained those services here in the molecular sciences main building. But the genomic, and there are still some equipment from the United Core that have remained here. But all the equipment for doing like microarray analysis or next generation sequencing were transferred physically to the Puerto Rico Comprehensive Cancer Center because they're going to be helping us to pay for the maintenance of the equipment and support and technical support. So that's the reason why we did the transfer. So in the molecular biology facility, I'm sorry if you don't see that so well, we still have the equipment to quantitate RNA, DNA, or proteins using the nano drop system. There's a new piece of equipment that is indicated there in red, the ABI 3500 genetic analyzer. The two Sanger sequencers we had, one had 1 capillary, the other one had 4. So we could only run a maximum of 5 samples per run. They were updated to get the 3500, which has it's an 8 capillary system. So now we can do more samples per run, 8 samples per run. Still, the other option was 24, but it really is not worth it. We never have that many samples at a given time. And also, you have to fill the 24 capillaries, so it would have been more expensive to do a run. So that's why we have the 8 capillary system. There are other accessory equipments for PCR, all kinds of PCR systems, both in the molecular biology core facility run by Dr. Rodriguez Medina and by myself. In mine, we have some thermal cyclers that allow you to optimize your PCR reactions because they have the gradient PCR capabilities. So we have 2, at least, gradient PCR systems that you can use to optimize your PCR. Once you set up the conditions, then you use the regular thermal cyclers. We also have equipment that has been acquired through the RISE program, through an equipment supplement. They're still all here. The Step 1 Plus, RealTime PCR machine, the EP Motion 5070, this is an automated pipetted system, which I highly recommend to anybody who is doing real-time PCR to do it using the automated pipette system. That way you minimize pipetting errors during the real-time PCR reaction. Somebody, you're setting up your plate, somebody talks to you, lose count of where you were, so if you do it with the pipetter, the robot doesn't make that mistake. So we also have the Odyssey Imager here. The other one that we originally bought through the RISE supplement was donated to the Neurobiology Institute because they were one of our highest users. Now, thank God, there's more users here in the campus, which has reduced the need for having a dark room and the chemicals and your codes being stained by developer. So the Odyssey Imager is a new system we bought 2 years ago. through the RISE program. And also we had acquired, but nobody's used, a mouse pod, which is for imaging using the same dyes that are used in the Odyssey imager, infrared range fluorescent dyes using animals. And so we have it if you need it. Okay. We also have a UV visible spectrophotometer that was acquired also with and a fluorimeter that was acquired to the RISE program. And they're all still being maintained and updated. So now the new unit that was created when we joined the RCMI and PRC-TRC programs for the Genomic Services, it's called Genomic Translational Research Unit. Now that facility is located at the Cancer Center. So that includes the Affymetrix gene chip scanner, which was recently updated. We needed to acquire a new computer because the one that came with the instrument was dead. So we have a new computer for it. We also had to acquire a new hybridization oven that Dr. Jorge Duconge kindly provided. So now we can run the more modern chips, gene chips. Oh, I have a show and tell. So I'm going to show you when I'm talking about the Affymetrix scanner, what are the gene chips. So here's the first one that covered the human genome, the U133 array. And here I have this is a gene array. So the 3' IVT arrays looked at the 3' end of messenger RNA. So the probes were designed for the end of the messenger RNA. The gene arrays do gene level detection. And there's also exon level arrays, which tell you which exons are being transcribed more frequently than others. So depending on what is your need of data, you decide which array to use. I think I have one for here. Which is another with 133. So the main types of arrays that we have done so far have been mostly expression arrays, gene and exon arrays. Dr. Vivas has used the microRNA arrays, and Dr. Duconge has been using the drug metabolism SNP arrays. These are genotyping arrays rather than expression arrays. The Affymetrix company has developed software for rapid analysis of those software. We also have the Partech Genomics Suite, which is a commercial package that allows you to analyze genomic data, both for microarrays and also next generation sequencing analysis. That's through the RCMI program. One of the key instruments that was acquired a long time ago for another purpose, but we eventually repurposed it for the genomics facility is the Agilent Bioanalyzer. This is a system that I recommend anybody doing any expression analysis of RNA should analyze their samples through the Agilent Bioanalyzer to verify if the quality of your sample is adequate enough and has the integrity values that are required for any downstream analysis such as real-time PCR, microarray, or RNA sequencing. We have let the genomics for in the Cancer Center borrow it, so it's still owned by the Medical Sciences campus, but we allow them to have it because if not, it's very hard to have a genomics facility without this instrument, which is also used for all the quality control steps in microarray or in next generation sequencing analysis required that you at different steps verify your sample using this instrument, different types of chips that come with that instrument. So this is a capillary electrophoresis system. I have here a sample of the RNA chip. It also has a cell analysis system, but I actually let Dr. Vivas use it. We haven't been using it. So this is an example. So this is a capillary electrophoresis system that is used for quantitative assays. Check amount and size of proteins, RNA, DNA, etc. So this is an image of the Affymetrix scanner, which even though we bought it over 10 years ago, it's still a relevant instrument. I guess it can't go to a smaller size for the spots where they put the probes for the arrays. So that's why it can't go lower than 5 microns. So that's why we still have that system. And it comes with the hybridization oven and also the washing station, which is used to process 4 arrays at a time. So at a given time, we can only do 4 arrays at a time, and then scan them individually in the instrument. these are the applications. You can find them in the Affymetrix website. There, it's a very useful website. It provides a lot of information. Okay, and we have the pricing information from the company. The next generation sequencers we have, there's two types. The ION personal genome machine was purchased by the RCMI program using funds from the Puerto Rico Science and Technology Trust, matching funds for the previous RCMI grant. And in this instrument, you can do targeted DNA sequencing, targeted RNA sequencing, the gene panels that have already been developed by the company or by users of this platform for either cancer, research, inherited diseases, both at the RNA and the DNA level for the cancer panels. So there's already lots of tools to do analysis using this instrument. So for small kind of sequencing projects, you would use the ION personal genome machine. For larger studies that involve like human genome, complete genome sequencing, human exome sequences, you can do 2 exomes per chip for the ion proton. I brought you here. Here's the ion proton chip. So here you would do 2 exomes. That would mean you have 2 samples, and you would label each sample with barcodes, and then you can analyze 2 different samples in one run in a chip. For the other instrument, the chips are smaller. So these are the ones that I'm passing. Let's pass it with the box. And you can also do whole transcriptome analysis, like RNA-seq would be done in the ion proton chip. Okay, and this is an image of the new Sanger sequencer. So remember, if you do a next generation sequencing experiment, many procedures require you to validate your results using the state-of-the-art sequencing technology, which is Sanger sequencing, which are more used to. This is the new instrument we bought also through the INBRE grant. The INBRE grant provided the funds for the new Sanger sequencer and also for this new real-time PCR machine. The only thing I want to tell you about this new machine, because we already have here a few in the main building. This one has an additional color for detection. The one we have here has 4 colors, a step 1 plus. That means one of the colors you use it for loading control, which is the ROX dye. Then you have only 3 for analysis. This one has 4 for analysis and then the 1 for ROX, for the loading control. This instrument is also qualified to do the real-time PCR assay, which is used for detection of simultaneously in one sample of chikungunya, dengue, and Zika virus. So it's either the One Studio system or the 7500, okay? And then this equipment is here in the main building. It's a kayak cube. There's also another one in the cancer center. It can be used to process small samples. And when I say small samples, I'm talking about 200 microliters of blood, or, for example, a dry blood sample to isolate DNA or RNA, or also small isolation of proteins, like, for example, his tag proteins. And the Odyssey imaging system, I already talked about it, so I guess a lot of you are using it for one of the applications, which is to look at, for example, total EGF receptor versus how much is phosphorylated. You can do the ratio by using 2 different antibodies that are labeled differently, either with a green dye or a red dye, and you can do the ratio of phosphorylated versus total. And that's the best way to do it. And as I said, we have the mouse pod, which nobody's used so far, and I hope somebody gets to use it. I'll let you now with Dr. Adelfa Serrano.
Good afternoon. I'm the director of the Tropical and Emerging Infectious Diseases. ¿Por qué se fue? Core. This is the only core dedicated to infectious diseases in the whole UPR system. And I think it's relevant because we live in a tropical country and have several infectious diseases endemic here, like Zika, dengue, and chikungunya, among others. Dr. Emily Colón Lorenzo is my assistant, and don't know this. Yeah, but it doesn't go in the core. And the goal of this unit is mainly to provide instrumentation facilities, space facilities, and some key service, especially for people working in tropical emerging infectious diseases, but we also support other investigators working in diseases relevant to Puerto Rico and to minority health. Our goal is to provide training in terms of everyone using equipment or designing experiments. We provide training, technical expertise, and support, as well as organize and coordinate seminars and workshops or symposiums that will help people to enhance their research capability in infectious diseases. We usually do this in conjunction with the neurobiology unit, as they have interest in infectious diseases too. We have done several in the past. And more importantly, in the new grant, we want to promote the integration of services and support among all the RCMI cores. Our facility is located here in the third floor, laboratory A349. Is highly used. In the past cycle, we have more than 8,000 uses, more than 2,000 uses per year. So it's a facility which really provides support to many investigators, students, and researchers here. We have a dedicated biosafety level 2 room equipment to work with infection agents and tissue culture. Some of the equipment we have is the Spectra Max multimode microplate reader. This is highly used, it's very versatile, and it's used by researchers from almost all the departments here. It has a visible light, fluorescence, and luminescence. We have also a pulsed gel electrophoresis system to separate big molecules of DNA, and also is used for identification of bacterial strains, or can be used for fungal too. The AMAXA system is a specialized equipment for difficult transfections. We have a fluorescent microscope with filter for red, green, and blue fluorescence, a gel dock available for everyone, and then basic equipment such as a medium speed centrifuge and a PCR Hood, etc., which people who are new in the system can carry out their experiments there until they have their feet in the ground and their own lab running. We also have a couple of programs which are very useful for cloning and design and for biostatistical analysis. And this is basic equipment. In addition to that, we provide training and consultation services, anti-malarial drug screening in vitro and in vivo. We have collaboration with Harvard and Broad Institute for this type of assays. Transfection, cell cytotoxicity, erythrocyte lysis assay, and most importantly, cell culture facility for infection agents. Through the year, we have supported more than 100 projects, 35 of them funded by external grants, numerous publications, And we keep collaboration with investigators in the infectious disease areas in different parts of the United States of the world, such as Johns Hopkins, Harvard University, University of Georgia, among others. Thank you, and I leave you with Mark.
Thank you, Adelpha. Thank you for inviting me. This part of the presentation is on the neurogenetics and bioimaging core, which is located at the Institute of Neurobiology. If any of you don't know where the institute is, you should not be looking for this sign because it's hasn't been there since the hurricane. But we're in the process of making a new sign. And really we'll update our RCMI panel at the bottom here. We're in Old San Juan, basically between El Morro and La Perla. The objectives of this cycle's grant is to enhance RCMI neurogenetics and molecular neurobiology core facilities with advanced bioimaging instrumentation and infrastructure, support programmatic activities, some of which have been mentioned already, the seminar programs that we do with the infectious disease, that will promote dialogue and collaboration with the other RCMI core facilities and between basic and clinical neuroscience communities. And finally, we try to stimulate national and international networking between investigators here and those addressing health disparities in neurological disorders elsewhere around the world. Okay, I'm going to tell you a little bit about the various resources that we have within this core. Okay, so it's the first one I'd like to mention is the RCMI Neurogenetics core. So you can see here we have incubators and other facilities. This grew out of the RCMI grant of 2 cycles previously, where we recruited a neurogeneticist, Bruno Marie, and this is Carolina Maldonado from Dr. Marie's lab, okay, working at and these are all fly stocks. If anybody has the need to use Drosophila, we have a lot of fly stocks and space for housing more. This is not my field, but one of the things that the people that work with the flies do a lot of is pushing flies around under a microscope. Okay, these are fly pushing stations. Okay, we have a couple. This is the RCMI technician, Luis Quiñones, and he's very skillful at doing this, and he could train anybody to do it. We also developed over the last several years a RCMI molecular neurobiology core. Okay, this is contiguous next to the neurogenetics core. It's got a few benches that really is open to more or less anybody to use. It's run by a committee, and anybody can apply to use benches for short or medium-term projects. We have, basically our instrumentation, PCR, RT-PCR. You can run gels, RNA, DNA extraction, quantification. In general, I would say that our instrumentation is not as sophisticated as a lot of the instrumentation that you have heard about so far. We really, in terms of sophistication, we specialize in the imaging. But people can get their projects started over at the Institute and bring, their samples over for some of these more higher end analysis. So that's the molecular neurobiology core. Okay, now I'd like to move into the imaging core, and it has a fairly long history. It started in 2004 when we purchased this instrument, which is a Zeiss Pascal laser scanning confocal microscope. Okay, a pretty early model of the confocal microscopes. Now, I think unhappily for Zeiss, our instrument's still operating now, like 13 or 14 years later. It's really been a tremendous instrument, and it still is relatively heavily used, and they don't service it anymore, but we've sort of managed to work things out on the island to keep it going, and I really like it. It's been a great instrument for us. Okay. Another instrument that we bought around 2008 is a live imaging facility in laser confocal scanning microscope. This is in the same room as the Pascal, sort of on the other side of the room. Okay, this instrument is less heavily used than our others, and I would be delighted if anybody has or can develop projects using, say, calcium imaging, calcium sensitive sensing imaging, or voltage sensitive imaging. This is really an excellent instrument. It uses a lot of Zeiss hardware interfaced with what's called a spinning disk technology developed by the 3i company, Innovative Imaging. And again, it's 2008, but it's still, it's still close to being state of the art and highly functional instrument. Okay, that's the live imaging facility. Okay, and then finally, this was about 3 years ago, we purchased a Nikon A1R laser scanning confocal microscope. This really is state of the art. This was purchased with a National Science Foundation MRI, Major Research Instrumentation grant that enabled us to remodel a room. A lot of you probably know these guys from Nikon, okay, basically the Rosado family. This is Amelia, who has served as the technician for this for the first couple of years. She was supported by the NSF MRI grant, and she's gone on to be a postdoc. But you do get technical support if you need to use this instrument. Now, one thing I would say briefly about confocal instrumentation, using this confocal, is we would really encourage you. You've seen regular fluorescence microscopes in some of the other presentations, and I really would encourage you, if you do this kind of histology, to have a look at your prep on a normal scope before you bring it to image on the confocal. I can tell you, I've been doing this for a long time, and if you don't see pretty good staining on a normal scope, it's not going to just show up because of this technology. Okay, so we really encourage people to use this for the publication quality images. Now, the previous one, the Pascal, if you really can't get access to a regular upright fluorescence microscope, come and use the Pascal just to see, the quality of your staining. We entered into a relation. This was about in 2015 with Nikon, where the University of Puerto Rico became a center of excellence. This has a lot of ramifications for all of you who want to purchase Nikon instrumentation with the price and the service that you can get, because the UPR is a center of excellence. You should definitely look into this. And then finally, I'd like to show you a couple of the results of the use of our instrumentation. This is an image of a Drosophila brain by Jonathan Blackburn. To recapitulate, we've expanded the use of the Drosophila model. In addition to Dr. Marie, Dr. Blackburn shifted into using this model, and a recent recruit, Dr. Andrew Seeds, is using the Drosophila model. So it's a growing model at the UPR. And finally, this image was not captured on any of our instruments. This is like probably taken with a regular camera of a squid, but this is from a paper of Josh Rosenthal that was highly visible in Cell, and it did have images in the paper that were produced in our facility. So thank you.
So good afternoon. I'm going to try to be quickly. So we are going to talk about the Integrated Informatics Services core. The overall goal of this core was to provide one point of contact to assist the researcher at the medical science campus with all the general aspects of the integrated informatics solutions. So the idea was try to give a place now related with informatics to go to get the services from the health informatics, bioinformatics, and operational informatics. This is the idea of this core. Was separated in 3 different informatics could be units: health informatics, bioinformatics, and operational informatics. So for the health informatics, Brenda is this is the staff now, also the core. Brenda is the responsible now, is in front of the health informatics. Brenda Nieves, Kelvin Carrasquillo in the bioinformatics, and in operational informatics, Alex Lora. We have also the support of Dr. Jose Conde, who was for many years, working on this in this area, and now he's our support also for this core. So we offer different kind of services, related all of them with informatics. In the case of health informatics, we offer the database management. So it could be like using the RedCap database for creation and consultation. When you have clinical data, then you can come now, and we offer the creation, consultation, etc. to create and manipulate this kind of data. But if you want to create a new database, like a relational database with some information you have, you can come over, and we also offer the development of the database, the design and everything for you with data. Or if you have already a database and you want to do some pre-processing on this kind of information, we also can offer these services using different programs. We all want to have our information, our research available on internet. So in that case, we also offer websites. So we are able to develop, design, and implement websites, host them in our servers, give maintenance to the websites, and update the websites. We use different programs to do so, like Drupal, WordPress, and when it's a little more sophisticated with databases, we use PHP, MyAdmin, etc. This is one example of the website that is already hosted by our facility, by our core, the Biomedical Information Unit. Also, if you need some software that you need to develop, and a specific software, we also develop tailored software. Like if you need like one specific program that you cannot find online or is not available, we can develop it. We do the design and implementation, hosting if it's a server client software, and we do update, etc. Different programs, language programming, we can use like Python, R, Java, C++, and this is an example that one new set of tools that we are developing right now in Python for ancestry analysis. The other kind of services we offer is bioinformatics. It was the joint of health informatics, bioinformatics, and operational informatics. In the case of bioinformatics, we offer the gene expression, gene or protein expression analysis using either microarray data or RNA-seq. So Dr. Cadilla was talking about different instruments that they generate this kind of data, so we can keep doing the analysis of this data. We already have done some analysis like differential expression genes and network enrichment analysis to breast cancer cells in treatment with isoflavonoids. Also proteome analysis. This one was with biochemistry department. We have done proteome microarray analysis to discover association of cleft palate syndromes in Puerto Rican children with a dental school, and also RNA-seq analysis to identify differential expressed genes in Arabidopsis thailand associated with spinal muscular atrophy. That was with New Mexico University. For the RNA-seq, we have already defined like a pipeline with different programs, like TOHAD, COFLINS, COFMARS, COFDIP, all of them from the bioconductor package. And in this case, we were working with plants, so we couldn't use the engineering pathway, it's more focused on human. So we used the cake mapper, that is like the engineering pathway, but for any species. And these all are free software. We also have done some microbiome data analysis using CHIME, so sequence, and come with the FASTA-Q. And then we did some study using that 16S to discover salivary microbial communities associated with obesity and periodontal disease in Puerto Rican adolescents. Also some SNP and admixture analysis, like variant calling analysis using these tools, population-specific ancestry, and locus-specific ancestry. So this is actually a current work that we are doing with Dr. Ducohen, and we are generating the pipeline. Other services, protein modeling and docking analysis. So if you have the data, come with us. For the protein modeling, we use programs like Fire2 and iTaser. And for the protein interaction dockings, we use PatchDock and CruisePro. Right now, we have already done a study of protein structure in yeast for discovery of antifungal treatments in humans. For cancer genomics, we have done some work also in database search and analysis from data, the cancer genome, the TCGA data, and the ICGC data, and using portal as interfaces as eBioportal to show and analyze the data. We have done some studies with Dr. Pablo Vivas related with ovarian and glioblastoma. Also for tumor driver gene identification, for somatic variant detection, analyzing between the tumor and the normal tissue. We're right now doing some studies in colorectal cancer. The other area, the operational informatics, we offer this service of software installation and server management. It's installation, update, upgrade of software, operating system, hardware. We also give first line technical support for computer connected to instruments and storage, cloud-based systems. Global communication service, we set up webinar through Skype for Business or similar software, and also record and upload videos in YouTube, like this seminar. It's being recorded and it's going to be uploaded to YouTube. Also, when we offer, the support for projects, if you are writing a project and you are planning to buy equipment and software in that project, you can come to us. We're looking for quotations, etc. You include in your budget some amount of money for installation, etc. So when you get the grant, we can install the computers, we can install the software, etc., and work with you with a warranty and these things. We also support all these services with the official lab, the Bioinformatics and Health Informatics lab. We have a group of students that also we develop research applying computer science to biomedical data, specifically like machine learning methods applied to physiological and genomic data.
Yeah, sorry.
sorry we went a little bit over our time, but I wanted to remind you that to acknowledge the program support, and I want to thank you everyone for being here. Also acknowledge if there are facilities with other support, like the location in the cancer center, or whether the PRC-TRC genomic instrument, also acknowledge their grant, etc. So all the programs are described here, the Institute of Neurobiology, the INBRE program, etc. So thank you very much for your presence, and we wanted to have questions, but I think that the time is taking over. I know you guys have classes, so thank you very much.